I'm Peter Black, urologist at the University of British Columbia in Vancouver. I wanted to give a brief summary of one session of our inaugural Bladder Cancer Forum in February. We had a session about translational research. So this is research that we take from the lab into clinic. And it builds on a session we had in April of last year where we got together various groups in Canada who are working on bladder cancer. And we identified 4 working groups that we've moved forward that involve collaboration of different researchers and clinicians across the country. And so the makeup of our session at the meeting was to bring in outside investigators and researchers to tell us what they were doing so that we can build on their expertise and potentially collaborate with them. The first speaker was Daniel Di Cavallo from the University of Toronto, who's a superstar in the area of epigenetics. We hear a lot about sequencing of DNA where you're looking for mutations and other alterations. Epigenetics describes alterations to the backbone of the DNA that doesn't change the sequence, but changes how the DNA is read and turned into RNA and proteins. And so his expertise is sorting that out. And we know that it's particularly important in bladder cancer because there are genes that control these processes that are very frequently altered in bladder cancer. So it's an enormous unmet need in bladder cancer. And Dr. Di Cavallo explained some of the mechanisms that they've uncovered in other cancers that clearly could be applied to bladder cancer. Our second speaker was Kathy McCoy from Calgary. And I think Kathy is a huge find for us as a bladder cancer community. She is director of the International Microbiome Center, a big center in Calgary dedicated to looking at the bacterial makeup of the gut and other organ sites, including the bladder. And we know that the microbiome is particularly important for how the immune system works. And Kathy was able to explain some very elegant work that she has done in colorectal cancer to show how the bacteria in the gut, she's been able to figure out exactly which bacteria in the gut make a difference. She's been able to show that these bacteria produce substances that they put into the blood that affect the immune system and the immune system's ability to fight cancer. So truly revolutionary and exciting work. And we're already trying to establish collaborations with Kathy. For example, there are mouse models of cancer where the mice have no bacteria in the gut, and you can add back specific bacterial species to see how it affects cancer formation and response to therapy. So the third talk was by Brad Nelson, who's in Victoria. He's an immunologist. You see that these 3 first groups actually come together around immunology, but he's dedicated to tumor immunology. And he spoke specifically about what makes some tumors hot, which means that they have a lot of immune cells in there on the prowl looking to get rid of the cancer, versus others that are cold and have no immune systems. And he's figured out some very exciting mechanisms that you can then also target with drug therapy. And so there's a lot of interest in that in bladder cancer as well. And then our fourth speaker in the session was Rodney Ouellette from Moncton, New Brunswick. He heads the Atlantic Cancer Research Institute, and his expertise is in what we call the liquid biopsy. So trying to figure out information about the cancer based on what's circulating in the blood. And he looks especially at what are called extracellular vesicles, which are little pieces of debris, essentially, but they carry RNA, DNA, protein, and other substances from the tumor. And so with a simple blood draw, you can see what's going on with the tumor. And it's especially interesting to do it over time at multiple time points while the patient is on treatment. So it was an extremely rewarding session overall, lots of opportunity for collaboration with each one of these investigators that should help move the bladder cancer field forward.